喂，对他换另外一只就是，哦，有看到了，
如果你没上过，
对，
就是连一些没有讲过要去。
好，那我选到，那声音的部分是用使用系统设定的还是线路设定
啊，
啊？对，设定就可以了。嗯，对对，三只都有就是了，三在四，四只，
对，现在是真的上面。
对，桌上也可以，其实无线跟桌上其实声音都会进去。好的，那Webex，对，有在动，嗯，OK，那应该声音有进去。
This meeting is being recorded.
不办便当
不不，自证，所以他现在是在录着他现在是在录我的那个投影片嘛？哦，确定哦。
看一下。下课再来录。嗯，
确定？你确定现在是我线上是我的投影片？
对，现在是你。
哦
我用手出来。
Oh, okay. Good morning, everyone. Happy New Year. So, my name is Minghan Cai, and the faculty from Institute of Immunology and Microbiology in National Yangming Chiao Tung University. And today I'm be requested to give you a principle for immunology within 3 hours. Let's see what will happen. So today my lecture will offer around 50% knowledge, around the 50% amount of knowledge from the general immunology. That is approximately encompassed from chapter 1 to 10. Yeah, so it will be a lot, but I will only speak something very important, but not speak in detail. So since it's an international school, so let me briefly introduce myself. And my name is Minghan Cai, and you can see my experiments are mostly focused on the immunology at the beginning, and therefore I switched to virology and immunology both. And currently, I'm now studying virology, especially herpes virus, Epstein-Barr virus, and humanized mice model. And now I switch to the virology, and especially the oncovirus, Epstein-Barr virus, and human carcinoma, and how to apply virus in biotechnology usage. Yeah, if you are interested, you can search my lab website. So here's today's outline of this lecture. And today, yeah, I will try my best to introduce as many things as possible in immunology in these 3 hours. And anyway, so I will want to give you a concept of immunology because immunology is I think it's the fundamental science of medicine. So no matter your major in biology or bacteriology, and I highly suggest you need to have background for immunology. So today's my concept is what is immunology? I will give you a brief history of immunology. Of course, everyone knows the vaccine of smallpox. And in brief, I will talk about innate immunity and adaptive immunity, and introduce the specificity of the human immune systems. For example, how immune system recognize self and non-self. And how our immune systems give us the diversity that can recognize as many pathogens as possible, and also how our immune systems get out the specificity that recognize the specific pathogens, and also the memory of our immune system. So let's go to the history. So no matter immunology or immunity, the definition of this word is from Latin word as well. So I believe tomorrow's biology lecture, I can also give you lots of Latin words from alpha, beta, gamma to omicron. And And the meaning of immunology is how our body protect ourselves from getting disease. And especially if the definition of immunology currently is the definition of how our body recognize self and non-self antigens. So pathogens, of course, they are non-self. And just before the modern medical knowledge, for example, medicine, immunology, biology, bacteriology, and I think currently you might hardly believe just before 115 years ago, people cannot live longer than 35 years old, believe it or not. So, of course, before that time, people relied on religions, and most of the time, if the disease outbreak, no matter bacteria or viral disease, people always claim, oh, we do something wrong and we were punished by God, whatever. But after the advanced knowledge of immunology and bacteriology and microbiology, and we can see that our life expectancy become longer and longer and longer. So we really need to think this knowledge. And the biology start a little bit later because biology requires lots of new technology because they are too small, you cannot identify. So here's so before the history of the immunity or immune response, when people reintroduce history immunology, we always start from smallpox. And the smallpox actually is a disease that had been identified for, I don't know, 30,000 or 40,000 ago, because from the Egypt mummy, yeah, he's the one in the movie, yeah, mummy. And here is the king of the Ramesses V king in Egypt. And thanks for the mummy, so we can see that it's quite clear this king perhaps died or he had ever suffered from smallpox disease, because you can see that there are many pups on his face. Yes, and he was buried in the King Valley. I visited before. I seen it. Yeah, and that is how the person alive when this young child suffer from smallpox, it looks very terrible. And usually, this kind of disease have extremely high fertility rate. Yet 30% people after getting this disease will die. And the very famous story is when the Spanish people came to North America and they deliver this virus to that place, and this smallpox virus killed nearly 500 million people. I want to say this virus killed 97% of American Indians after the visit of Spanish people. And of course, this disease is caused by a virus called smallpox. And therefore, there's a person called Edward Jenner, and who was identified as the father of modern immunology and the vaccine. So let's see how this person performed before. So, of course, smallpox is a very terrible disease, and that killed lots of people every single years. But thanks to the Edward Jenner observation, he observed that the milk maid means the woman who collects the milk from the cow, and these ladies seems very difficult to get smallpox infection. But on the other hand, it seems these milkmaid and quite easily to suffer from another kind of mild disease that was transmitted by the male cow, and we call that cowpox. So according to his observation, he found that if a person that was infected by the cowpox, then therefore this person will get immunity against the other virus, another disease caused by smallpox. And furthermore, Edward validated his hypothesis by doing experiment like what we are doing every day. Yes, and he vaccinated or he inoculated the small cowpox from cow into the young boy, and therefore after the so-called vaccination, he rarely inject the smallpox into this young boy, and fortunately this young boy survived. So that is how people validate the function of vaccination. So after his validation and many further experiments, and Edward Jenner eventually convinced the government of Great Britain, British Empire, to let the inoculation of cowpox into the general citizens of Great Britain. And you can clearly see that this table chart represents the death per million inhabitants in Great Britain, and according to caused by smallpox infection, you can see that every year around 3,000 to 4,000 people die at average in Great Britain. But you need to remember Great Britain was the strongest country during that time. And after the compulsory or introduced vaccination by the British government, you can see that the death caused by smallpox deeply dropped to nearly zero. So please understand the importance of the vaccination. And you need to understand how this story was happened before Pasteur found that human disease caused by bacteria. Everything happened before that. So no matter how, we still need to go further for the history. Another interesting history is the emperor in Qing Dynasty in China. And believe it or not, there are actually quite many emperors died due to smallpox infection, quite frequently at the early time point in Ming Dynasty and Qing Dynasty. So the third emperor of Qing Dynasty, called Shunzi, actually died due to smallpox when he was just 22 years old, very young. So at the time before he died, and he asked one very good friend from Germany called Adam, and he asked, if I died, who should after me become emperor? And these German people tell him, you should give the emperor position to your third son because he had survived after smallpox. So you don't need to worry that your third son, after becoming emperor, he will die unexpectedly in the soon future. So that is the reason why the fourth emperor in Qing Dynasty, called Kangxi, became the emperor thereafter, because he survived after smallpox. Smallpox is deadly, but not all the people die after infection. And from the ancient people, they already know if you survive after smallpox infection, then you will get lifelong immunity against this kind of disease forever. So there's another interesting story. Actually, this Kangxi emperor was also the person who introduced the so-called vaccination strategy in the Chinese population. And in brief, and he introduced, and the people will collect the pumps from the person who has survived after smallpox infection. And of course, this virus get compromised from this survived patient because their immunity or antibody already combat this kind of virus. And therefore, the people will collect this material and use a tube and to blow inside to a healthy individual's nasal cavity. And by throwing this weight, believe it or not, and the smallpox epidemic in China dropped significantly to less than 1%, I remember. And this technique also be introduced into Turkey and Russia, and therefore delivered to Europe. if you see the time point very carefully, actually this time is 100 years earlier and Edward Jenner. So anyway, these people did lots of good things for human population. So that is the story. So it's very cool. So immunity, or we call vaccination, can help us to combat smallpox, these very deadly pathogens. But believe it or not, smallpox actually also kill human beings through immunity. For example, let's just go through the pathogenesis of this virus very briefly. So this virus is very highly transmissible because it can be transmitted through aerosol, like COVID-19 right now. And after that, it can replicate in the lung and respiratory tract, and therefore go to lymph tract and delivered by macrophage, and therefore go to everywhere. And after that, it will cause a severe hemorrhage through to very strong inflammation, and therefore people die. So actually, this disease is caused by our immune system behave very strongly. On the other hand, I can give you many good examples to tell you that immunity is actually a double-edged sword. For example, Ebola virus is also very deadly, right? I think its death rate is nearly 90%. And this virus also kills people by overimmunity because it can also cause very, very strong inflammation in the endothelial cells. And therefore, all endothelial cells go into inflammation and become very leaky. So all the body fluid will flow outside. So people died very, very terribly because all the body fluid will come out from eyes, nose, ears, everywhere, every single hole of your body. So overimmunity. And the Spanish flu in 1918s that killed nearly 100 million people at that time, 100 years ago. And according to the modern science published in Nature, and they try to recover this deadly virus, and they validate this Spanish flu virus was so deadly, also caused by overimmunity, because this virus prefer to kill the people have good immunity, but not the young children or the old people. Very different from our seasonal flu virus. And COVID-19 also caused severe disease due to cytokine storm. I think you had heard about these terms quite frequently from news or from paper. And dengue virus also caused severe disease due to antibody-dependent enhancement. I think you hear antibody, you know it's associated to immunity. So actually, many diseases we suffered annually actually caused by too strong immunity. So very strange. On the other hand, and there is another kind of disease called autoimmunity, means immune system attack ourselves. And if you remember, our immunity is the key point of immunity is recognize self and non-self. So when the immunity have problem, immune system have problem, when they have chance to recognize our self-antigen, our self cells or tissues, and therefore these immune cells will attack our body and cause many different kinds of disease. For example, when immune system attack the insulin producing cell beta cells, and the people will suffer from type 1 diabetes. And when they attack the joint, then we will get the rheumatoid arthritis and the like photo. This one is quite frequently happen in the aged people. Very, very painful. And of course, immune system also have chance to attack neuron. And when the multiple sclerosis get to the end stage, people will die because they will eventually attack brain. And of course, there's another systematic autoimmunity disease like lupus, and one very suffering disease called inflammatory bowel disease. When immune system attack your gut, is very painful, believe me. And there are many, many other examples. So it's quite strange. Immunity should protect us, but there are so many people die due to immune-associated disease. But on the other hand, of course, everyone knows immunity is so important. We cannot even survive without that. One good example is when people get infected by HIV virus, and ultimately all the CD4 T cells be killed by HIV virus, and this patient will acquire something called AIDS. And when the symptom called X, this patient theoretically can die due to any kind of infection, no matter bacteria or virus. And on the other hand, we also learned lots of inherent immunodeficiency disease due to genetic defect. For example, there are quite many systematic severe combined immune deficiencies caused by inherited gene problem. And if people have such kind of disease, perhaps just 50 to 60 years old ago, these people should be negative selected because these people have no chance to survive in this world. But thanks to some advanced technique, so this patient now have chance to live in such sterile cabinet, and they are called bubble babies because they cannot expose to any kinds of bacteria, any kind of virus outside, or they will die just like AIDS patient. And of course, we know lots of knowledge that most of the immune cells are generated from our bone marrow cells. So now we know bone marrow transplantation might have a chance to cure these people. So just remember, actually, I believe all the people here, our immune system should be nearly identical to the people just 300 years ago, right? So we think immunity help us to protect from smallpox. Actually, is not correct. Actually, we manipulate our immune system help us to protect from smallpox infection. Our immunity is not perfect. So I will say it's something more like homeostasis. So no matter our immune system go too strong to the pathogen or too strong to our self-antigens, they will have different kind of disease. For example, when they respond to our own cells too weak and immune system too weak, and therefore we will suffer from cancer. And when our immune system are too weak, then of course, no matter bacteria disease or virus disease can come into us. And on the other hand, when immune system are too strong to outside antigen, we might get allergy. And of course, also many examples for virus disease. And when we immunity are too strong to ourselves, we will get autoimmunity. So you can see that no matter our immune system go to which way, you will anyway get disease. So anyway, today we are going to try to understand immune system as many as possible. So whenever time, if we get tumors or cancers, we can try to skew the homeostasis back to the central part. Understand? So our immune system is not perfect. And I think you might hear many stories about some ancient heroes. And perhaps there are many heroes who never got wounded all his life during his life, 100 rounds of battle, no wounds. But this hero might die just because he cut himself during cooking. Because just a small wound before the discovery of antibiotic might kill a person. So during the historical battlefield, actually people are lazy to distinguish you are wounded or dead. If you are wounded, actually you are dead. Before the discovery of antibiotic. Because I will tell you lots of information how our immune system combat bacteria, but you might wonder why people die so easily when there's no antibiotic. So how to remove this? So what are the key some concepts of immunity or especially for our human beings? Let me repeat again how the immune system recognize self and non-self, and how immune system get us specificity of different antigens, and also diversity allowed us to recognize as many kinds of antigen as possible, foreign antigen as possible, and most importantly, memory that allows us to invent the vaccine. So one very short question, whether only human beings or mammals have immunity? We have no time, of course not. So let me give you some many examples. Even bacteria, they have immunity. Bacteria sounds very strong, right? But actually, bacteria have a very deadly natural host called bacteriophage. That is a virus, DNA virus. It can easily kill bacteria. So anyway, during evolution, bacteria acquire some knowledge, or perhaps just due to evolution, some bacteria equipped with some restriction enzyme. If you have lab experiments for molecular cloning, you might know BNH1 called forgot. ECOA1 called GATTC, right? Is a specific DNA sequence. And this restriction enzyme for what? Attacking DNA virus, bacteriophage. And the other thing is very famous called CRISPR-Cas9 system or CRISPR-Locut. It's also acquired from bacteria. They acquire some foreign DNA fragment, especially from the bacteriophage, into their genome as a locus, and therefore express these guide RNA in combination with Cas9 protein that allowed this complex to digest the specific DNA sequence recognized by this DNA fragment and the bacteria collect. So the restriction enzyme CRISPR system are actually how bacteria immune from phage. Yeah, but it seems phage always win because they mutate faster. And therefore, after bacteria, therefore, we go to the low-grade multicellular organism, especially referred to in invertebrate animals. So of course, the pathogen they meet are usually, don't know how to remove these things. Go away. Anyway, so the major pathogen they meet are still bacteria and virus. Of course, they might not, like human beings, have blood cells and blah, 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 that can have a spatial body system that help us to keep immunity, but they have many different ways. For example, they have very hard skin, keratin, very hard keratin. It's quite easy to understand, just don't let the foreign things enter our body. Mucus, get away, don't enter my body. And some organisms also secrete some protease or lysosome or antibacterial toxin or peptide to kill the bacteria outside. And also there are quite many proteins that might recognize the pathogen surface structure. I will mention that later. And they also acquire some very innate immunity, like human beings, through enveloping similar pathway, or some protein that can modify foreign DNA and so on. So actually, this story I will mention again in human beings. So just let you know, actually, during evolution, nearly all the organisms, no matter they are bacteria, they have vertebrate or not, They all have their own immune system. And here is the immune cells, how in our body, and we will mention that later. Most of these are the cells used for innate immunity to combat bacteria or fungi. And if you compare to these human innate immunity cells to these very primitive multicellular organism, you can see actually they look quite the same, especially like the macrophage. They have many granular insides and they can digest the pathogens they engulf in. And of course, these invertebrate organisms also acquire some protein, and they have very specific structure, can recognize the surface of different kind of pathogens, especially bacteria. And in human beings, we call that toll-like receptor. But actually, this kind of protein you can already find in many low-grade organisms. They already acquired this system, and we will mention that also later in human immunity. So after when the animal get the vertebrate, and they have bone marrow and they can acquire the blood system, and therefore the immune system from the multicellular organism into the blood system. So therefore these animals can have the chance to have the innate immunity and also the acquired immunity. So what means acquired immunity or so-called adaptive immunity? And as we frequently know, this immunity means T cells and B cell immune response. T cells can modulate whole immune system as well as killing the pathogen infected cells or the strange cells. And B cells can generate antibody that directly conjugate to the foreign antigens or the foreign pathogens. So actually, all these criteria I mentioned again and again are the feature of adaptive immunity. Of course, innate immunity can also recognize self and non-self, but very, very permanent. Adaptive can recognize very specifically. So anyway, if our body have problem for immune system, as the story I said, many ancient heroes died during cooking. And if the bacteria have chance to enter our body, and therefore you can consider our body as a cell culture dish, and the bacteria will be very happily growing inside. So actually our body beneath the skin, keratin, actually you can consider our body is a sterile cabinet. And it shouldn't have bacteria inside our body, especially blood system. I mean inside our body. So what is the difference between innate and adaptive immunity? Innate, of course, means we already acquired naturally from our ancestor, and they need to do response very quickly. So they recognize the spatial structure we call PAMPs, or the other structure or receptor or protein or glycoprotein offered by pathogens, especially bacteria. And in our human beings, the innate immunity also participate in the connection between innate and adaptive immunity. Continue. So if our body need to have a functional adaptive immunity, also need a functional innate immunity. I will mention that later. So it is quick, robust, fast, and strong, but this system do not have memory. But for adaptive immunity, contains all the features I mentioned again and again and again. They have high diversity, high specificity, and also memory. But the disadvantage is adaptive immunity need more time to happen. So just give you a very brief example. when the time after we get wound and when the bacteria enter our body, at the first time, the innate immunity cells like macrophage, dendritic cell, neutrophil will firstly go to that place because they are usually reside beneath our skin. And when they get damaged, and these cells, epithelial cells or endothelial cells, they will offer some dangerous signal and painful, and they will recruit some innate immunity cells go there to eat these pathogens. And when these innate immunity cells meet these pathogens, so-called dangerous signal, they will cause inflammatory response, and therefore elect the activation of adaptive immunity. So of course, most of the small wound might be cured simply by innate immunity, but eventually adaptive immunity will also be activated. Because these innate cells, after acquire these pathogens, they will go into the lymph node and therefore offer these antigens to the adaptive immunity. For example, T cells and B cells. So again and again, self and non-self dangerous signal. So let's move to, I already hit this thing. Let's move to the our blood system. And all our immune system cells, immune cells in our blood actually all derive from bone marrow. So that's why only vertebrate animals have this kind of system. Invertebrate won't have this kind of system. So after that, the bone marrow will derive to common lineage progenitor, and they can be differentiated into either B lymphocytes, T lymphocytes, or NK cells. And some part of the dendritic cell also involved in this kind of lineage. And on the other hand, the other kind of lineage called myeloid cells, blood cells. And they can be differentiated into different kinds of the cells participate in innate immunity, like the neutrophil, eosinophil, basophil, and mast cells and monocyte. And monocyte can further be activated into macrophage. Macrophages cells really like to eat. And when we start everything from the beginning, we talk about innate immunity first, because only after innate immunity, adaptive immunity can happen. And NK cells is the cells between innate immunity and adaptive immunity, and I will mention that later. So which kind of cells in our innate immunity? Let me briefly introduce. Macrophage, dendritic cells, neutrophil, eosinophil, basophil, and mast cells. And each of them has different kind of function. For example, mast cells engulf the foreign antigen or foreign pathogens. And after that, these cells can digest the thing it engulfed in, and therefore digest them into pieces, and therefore present them as antigen on the surface, called major histocomplex. What happened? 我要选哪一个？啊？大家好，谢谢。Anyway, so even after these cells eat the pathogens or antigens, they will deliver these antigen pieces to the central lymphoid organ like spleen and lymph node and activate adaptive immunity. And dendritic cells is also the cells really like to eat something outside, and similar as macrophage, but they eat the small pieces of foreign antigens into their body and also present as antigen pieces on the surface and therefore go to lymph node to activate T cells. And neutrophil is the cells that have very lots of granules that can kill the bacteria, especially, and when they inject these granules into the bacteria cells. And these two cells called eosinophil and basophils, actually, and also mast cells, actually, they are mostly be used for protecting ourselves from parasites. I think due to the modern science, I think now very, very few people actually suffer from parasite, right? So perhaps we are not very familiar with it, but actually cells participate a lot in allergy reaction because parasites require IgE anti-hypertrophic response, and also the asthma and the allergy also use these two immune pathways. So, but anyway, we'll mostly focus on the cells here. What? Yeah. And just let me remind you, our ancestors, when they have no vertebrae, they already have a system to recognize foreign antigens. So what happened to human beings? We have more than that. We have the protein or the immune protein that help us to recognize the foreign antigens, or we so-called dangerous signals, called toll-like receptor. And currently, there are 11 different kinds of toll-like receptor have been identified. And these are the surface protein or the cytosolic protein inside the immune cells. And here, this table gives you some overview. For example, you can see that I think you have already made lecture for bacteria. So you can see that toll-like receptor 1 and 2, these heterodimers mostly recognize the bacteria cell wall or the spatial structure of the bacteria, like lipopeptides, blah, 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 for gram-positive, gram-negative, blah, blah, and fungi. So when immune cells, these protein or toll-like receptor protein, when they attach to fungi or bacteria, they will get crazy. And they will want to eat these bacteria and kill these bacteria. And this kind of toll-like receptor you can find in monocyte, dendritic cell, mast cell, eosinophil, and basophil. And therefore, we move to toll-like receptor 3. They recognize double-strand RNA, and this kind of thing you can find in virus. And this can be equipped in natural killer cells. And toll-like receptor is very, very commonly used and very famous. They recognize lipopolysaccharide, gram-negative bacteria. And they also equipped in many, many innate immunity cells, toll-like receptor 5 flagellin, the tail of the bacteria, and toll-like receptor 7, single-strand RNA for the virus, in the many different kinds of cells, immune cells, toll-like receptor 8 virus, toll-like receptor 9, DNA with unmethylated CpG methylation, because our human mammalian cell DNA are heavily methylated by CpG, but not happened in bacteria or the non-generated virus. And the toll-like receptor so far is not very clear. And 11 also recognize some spatial structure for bacteria. So you can see that our body actually have equipped many different kind of sensor that help us to recognize these bacteria. Because bacteria is outside of the cells. Not frequently enter the cells. So this receptor mostly located on the cell surface. And by location, you can see that this toll-like receptor, they can either locate it on the surface to recognize the spatial structure, or they can also become the intracellular receptor. For example, if we want to recognize the intracellular DNA, then you require toll-like receptor 9. And just give you some example. So actually, our innate immunity, these cells equip many, many of sensors for this so-called dangerous signal. Dangerous, very bad. And I think after the innate immune cells or the cells equipped with this toll-like receptor, they will cause, and the downstream of this toll-like receptor usually will generate different kind of cytokines, such like interleukin-1, interleukin-6, and TNF-alpha, and so on. And these cytokines are highly associated, are very, very strong inducer for inflammation. So what means inflammation is the symptom that your body will have heat, pain, redness, and swelling. Of course, and it should be quite easy to understand why the immune system want to elicit this thing. For heating, it can increase your metabolic, and also the heat can also reduce the replication of the virus. And pain, of course, is painful. And red is because this inflammation increase the dilation of your endothelial cells that allowed the immune cells squeeze out from your blood vessel into the wound tissue. So it will become very red because the blood vessel permeability become higher. And because, of course, if the cells can go out of your blood vessel, then the tissue can also go out into outside of your blood vessel. So it will cause swelling because the body fluid also go out. So that is a very general idea. When the innate immune cells recognize this so-called dangerous signal by toll-like receptor, it will cause inflammation locally. And there are some also different kinds of the receptor identifier associated to the recognition of the dangerous signal I list here. If you're interested, you can read it carefully. And this dangerous signal we call pattern recognition receptor. And they recognize PAMP, what means PAMs. Pathogens associated molecular pattern. So they recognize a specific pattern. Bacteria is quite easy to recognize. So it will be quite interesting because this is part in biology, but we can just briefly say. And actually, nearly all the virus that can cause human disease have, I don't know, at least several kinds of ways that can prevent from the recognition of immune system, no matter innate immunity or adaptive immunity. Because it's quite easy to understand, the virus can only survive after they get into the cells and they replicate, have more virus infect more cells. If the immune system can so easily recognize the virus, then this virus will disappear. It will not happen now. So while the bacteria don't get evolution into the way that prevent from our human immune system, because bacteria can live outside our body if there is sufficient nutrition. It's very different from the virus. Understand? So we don't need to so worry. The bacteria will have some mechanism that allow them to prevent from the recognition of these innate receptor. Bacteria, you can culture them outside in the shaker. Virus, you can only culture them after infecting the cells. So if the virus that need to get baby still survive in this world, the only ways they need to find a way to get away from immune system recognition. So these are very two different pathogens. So it is a very complicated signal pathway. I will not mention very significant in detail, and I offer many figure legends for you. But in brief, after the activation of this pattern recognition receptor or so-called toll-like receptor, and the downstream signal can lead the transcription of many inflammatory cytokines, and such like interleukin-1, interleukin-6, in the TNF-alpha, and therefore induce inflammation that require for the killing or cleaning out of these pathogens. And besides toll-like receptor or the other receptor that recognize the pathogens, there are more this kind of pathway be identified. So let me just give you some example. For example, the RIG pathway is very famous for the activation of immune response against RNA virus, because COVID-19 is an RNA virus. Because it's too small and too complicated. I don't know why immunologists also always like to make things very complicated. So I make things very bigger. So when the RNA virus enter the cells, And because many viruses, the RNA are not generated inside the nucleus, so many of them actually don't have 5' capping. I will show you what is capping later. So this spatial structure can be recognized by RIG-I. And because RNA virus, especially single-strand RNA virus, when they want to replicate, they need a minor-strand RNA viral genome be generated, right? So they will have a period when this viral genome have double-strand RNA. And these kind of structure are very sensitive for RIG-I activation. No time to explain. So just give you an example, COVID-19. COVID-19 is an single-strand RNA virus. If you want to have its genome replication, of course, you need a minor strand viral genome as template. So when the generation of the minor strand, of course, they will have a double-strand RNA. Just give you an example. So what is capping? So for the eukaryote cells, when the RNA are transcribed and processing in the nuclear, actually the RNA will have processed through splicing and adding the poly-A tail and also the 5' capping, like this structure. It is how the RIG-I recognize self and non-self RNA. And there's another mechanism also be found recently called cGAS-STING pathway. And it is a pathway recognize the intracellular DNA. And of course, our body have a nuclear membrane. And our genome DNA are actually inside the nucleus. It shouldn't happen in the cytosol, understand? So that is why it makes sense our body have this kind of system that recognize the cytosolic DNA. So what might be cytosolic DNA? DNA virus. Or some pathogen that have chance to enter the cytosol. And also this pathway is extremely important for tumor because tumor cells, their chromosomes are not in line. For example, our body have 46 chromosomes, right? In tumor, maybe they have 46, 48, 49, whatever. They are not aligned. So frequently, this small chromosome will be kept outside the nucleus during mitosis. And it can be also recognized by cGAS-STING pathway. So this pathway is also frequently damaged in tumor cells because this pathway is actually very strong. What now? So okay, after the inflammation, what will happen next? How to make this thing disappear? So after inflammation, what will happen? Red, swelling, pain, heat, yes. And the tissue damaged place will cause this kind of inflammation response, and that will therefore recruit the furthermore innate immune cells like macrophage and neutrophils go to the place that get damaged or get inflammation. So how they do that? So of course, the damaged tissue, epithelial cell, endothelial cells, you need to understand epithelial cell and endothelial cell, they can also offer the dangerous signal because they are very painful. They can also secrete cytokine. And anyway, in this place, they offer lots of dangerous signal, and they can also offer something called chemokine. Chemokine means the protein that can attract the cells that equip with the receptor of this chemokine to the gradient, to the concentration of this chemokine at higher concentration. So anyway, when the damage or inflammation happening here, the cells, neutrophils will be recruited to the inflammatory site. And remember, inflammation will cause the dilation of endothelial cells that allow these cells to squeeze into the damaged tissue that contain the higher concentration of chemokine. That's called diapedesis. Very difficult to pronounce. So it is on for not only the chemokine, when the tissue get damaged, and actually these kind of cells also have lots of the surface protein we can call selectin. So when the body is peaceful, no damage, and most of the cells will recruit with the E-selectin that only have low affinity binding to these innate cells. So the cells inside your blood vessel will rolling through your blood and go to your body everywhere. They will not be trapped here. But when the time this region get inflammation, and they will change the surface protein from E-selectin into another one and containing ICAM1. And this one have very, very strong affinity to another receptor of the innate cells called LFA1. And this binding allowed the strong interaction between the immune cells to the damaged tissue in combination with the chemokine, and therefore it can help these cells get into the correct place that have inflammation, have higher amount of chemokine, have damage, have dangerous signal, and allow these innate cells to kill these pathogens. So inflammation is important. And here I gave you lots of these selectin or the integrins that help different kind of cells get into the correct place when the dangerous signal happen. It's too complicated. We will not go through that. If you are interested, you can read. Okay? You have exam in the end? Oh God. Okay. So let's overview the innate immunity. So actually, if our body is strong enough and if there are not so many pathogens get inside our body, actually innate immunity can solve the problem. But anyway, the signal will nevertheless be transferred to adaptive immunity. We'll mention later. So let's go through what happened when our body get wounded. Firstly, the damaged epithelial cell will also release signal. Innate immunity not only offered by the blood cells, many of our body cells can also offer immunity. So these damaged epithelial cells will recruit macrophage, as the story I mentioned here. And the macrophage will recognize these PAMs, or dangerous signal of the pathogens, mostly are bacteria, and macrophage will get crazy and start to eat the pathogens that these macrophage can recognize. And therefore, the damaged tissue and the crazy innate immune cells will secrete lots of inflammatory cytokines that recruit more immune cells get to the damaged place. And of course, inflammatory response, pain, swelling, heat, blah, 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 allowed more immune cells get to the damaged tissue. And these immune cells will happily eat there, get crazy, eat, and digest bacteria inside, eat. And therefore, the macrophage and the dendritic cells, as the cells so-called antigen-presenting cells, will present these digest or the collect antigen fragment into the central lymphoid. I see something disappear. Oh, maybe I answer later. Yeah. Okay. So anyway, the macrophage cells will transfer these so-called antigen fragment to lymph node and stimulate T cell response and stimulate adaptive immunity. And we will also mention some other innate immune system that are not acquired by cells later, called complement. And of course, the damaged cell will secrete lots of signal like interferon alpha and beta that can protect the surrounding cells, also not be attacked by the pathogen. So how the innate immune cells, specifically neutrophil macrophage, how they kill the digest pathogens, and actually there are lots of material inside the endosome or lysosome of bacteria that can digest the eating stuff into pieces by acidification and toxin oxygen-derived product, toxin nitrogen, antibacterial peptide, enzyme like protease, lidozyme, blah, 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 and competitor. Yeah. So just believing they will eat bacteria. And here is the signal pathway of the toll-like receptor. I think to overview this thing is too complicated. So just trust me, this toll-like receptor will produce cytokines, interferon alpha, beta, blah, 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 that promote the inflammatory response. And just give this concept. And immune resistance are always associated to loss of signal pathway that make people very crazy. So I just gave you some example how these the cytokines receptor look like, and the downstream signal belong to which kind of pathway. And I also gave you lots of chemokine factors and the chemokine receptor, which chemokines recruit produced by which cells after getting crazy, and when they're getting crazy, produce this chemokine, they will recruit which kind of cells. It's too many things need to memory, so even within the exam, believe me, if you're interested, you can recite this table. And okay, so what happened after the initiation of innate immunity? For example, when the macrophage get crazy, and this macrophage not only eating and digest the eaten pathogens like fungi or bacteria, they also get into a crazy mode by secreting lots of so-called cytokines that can further contribute to inflammatory response. Just give an overview: interleukin 1 beta, TNF alpha, interleukin 6, and the chemokine 6CO8, interleukin 12. And you can see, if you read this table very carefully, you can see that actually some chemokines, some cytokines secreted by these innate cells actually connect to adaptive immunity. For example, you can see that interleukin 12 can activate natural killer cells, and also can induce the differentiation of CD4 T cells into T helper cells, T helper 1 cells. Oh. And I think you understand what that means in the later part. I just want to tell you that after the activation of innate immunity, it will also contribute to the activation of adaptive immunity. Interleukin 6 is a quite interesting inflammatory cytokines because it can be found in many kinds of cancer, and blah, 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 and all the others are associated to the inflammatory response: heat, swelling, pain, red. So after talking about the innate immunity offered by the cells, now let's get to another system. They are not cells, but they can also offer us innate immunity, especially against bacteria. We see our body has so many things to against bacteria. Just give you some example for the antimicrobial peptides or proteins, such like defensin, and they can target either gram-positive or negative bacteria, blah, blah, blah. And they have very strong positive charge, can directly bind to pathogens, and therefore make pores. When bacteria make pores, they will die. And there are also another protein that can stabilize defensin, and in oral cavity, we have another protein have similar function, and C-lectin can also kill gram-positive bacteria. And our body can also secrete some protein or enzymes that can directly digest the bacteria called lysozyme. So it seems our body really hate bacteria. And the other thing is, if you remember, I mentioned interleukin 6 is a very interesting inflammatory cytokine because it can also stimulate our hepatocytes to secrete more anti-microbial protein. For example, the surfactant A, B, and mannose binding lectin, blah, 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 blah. And all of them also have a strong effect for killing pathogens. But of course, because it is secreted by liver hepatocytes, so it's within our body. And the other things are mostly extracellular. They really hate bacteria. And finally, another innate system called complement system, also not cells. And this complement system actually can elicit very, very strong immunity. Actually, there are many, many different components of protein. And this protein can also recognize the pathogens by using PAMs. I mean the pathogen structure, blah, blah, blah. They have a specialty to recognize these bad pathogens. And after that, they would get cells assembly into a complex, and eventually they will make a hole. And this hole will permeabilize the bacteria and they can die. So you can see that some spatial structure can elicit complementary immune response, mannose, glucose, and N-acetylglucosamine. Oh, so long. And this complement can also have a very, very strong reaction in the combination of the immunoglobulin antibody. We will mention that later. And of course, this complement is also very important for the stimulation of adaptive immunity. And just give you some example how this complement connect to adaptive or innate immunity, because our immune cells have many, many receptors that can actually recognize the pathogens that bound to the component of this complement component. For example, CD35 recognize 3C3B or C4B1. And these kind of and can elicit strong immunity, and these cells can be found in many, many different kind of cells. So innate immunity actually can further stimulate innate immunity as well as adaptive immunity. You can find that I give you many, many examples because I know you will not buy the book. So just read the slides in your future life. And of course, because complement is too strong, they can cause very, very strong immunity. So actually our body also have a negative regulator of complement system. It makes sense. So of course, so there are quite many strong inflammatory associated disease associated to the deficiency of this so-called negative regulator component. And you can just read that. But anyway, they have very strong inflammatory outcome. Oh God. So I use 1 hour finishing immunity. Okay, so let's move to adaptive immunity. So let me tell you again and again and again the concept of the beauty of our body immune system is the ability to recognize self and non-self, and adaptive immunity offer this in more advanced version. And specificity, diversity, and memory. So when we mention adaptive immunity, it means lymphoid, lineage, T cells, B cells, and all NK is partially adaptive immunity. I will mention NK later. And the T cells, they have CD4 and CD8 T cells. And CD4 T cells is a key regulator for whole our immune system. Very important. Without CD4 T cells, CD8 T cell will also malfunction. And the CD8 T cells can recognize the cells that offer antigen and directly kill it. And B cell generate antibody. So before I think I speak too fast. Before we start innate immunity, so how many of you have ever learned immunology before? I mean the 4 lecture before you. Okay. All of you, so why I need to have this lecture? Oh, so it means I can ask question. So anyway, if I want to have an introduction of adaptive immunity, I need to mention many components. Just give an overview how human T cells be activated. Firstly, the antigen potential cell, short name called APC. They can offer the foreign antigens or even self-antigens into pieces into the peptide form, and therefore be presented by MHC complex. And this MHC complex, along with antigens, can be recognized by T cell receptor, class co-receptor. If this co-receptor is CD4, and therefore it corresponds to MHC class 2. If this receptor is CD8, then it corresponds to MHC class 1. It's a first signal, antigen presenting cell. And the second is the co-stimulation signal. When the antigen presenting cell acquire antigens and also be stimulated by the so-called dangerous signal, they can elicit some co-stimulation signal that help T cell activation. The most widely used called B7.1 and B7.2, CD80 and CD86, and the corresponding co-stimulation molecule on T cells called CD28. And here's the second signal. And the third signal presented by antigen presenting cell is cytokine, and this cytokine can decide the fate of the T cell response later. T cells can be differentiated into T helper 1, T helper 2, T helper 17, TFH, and the Treg. Yes. So before we tell about whole this story, we need to tell each component one by one. Antigen presenting cell, MHC class 1, class 2. What is antigen? What is TCR? And why TCR can offer diversity, specificity, and recognize self and non-self? And how the co-stimulation molecule be activated through antigen presenting cells. And for B cell activation, quite similar to T cells, but their antigens are not presented by MHC molecule. The B cells receptor actually is a surface bound antibody. And this antibody can be therefore secreted by this activated B cell, therefore into the soluble form. So it makes sense the antigen need to be recognized by the surface antibody, and therefore they can generate an antibody that recognizes this antigen thereafter. So B cells, in contrast to T cells, they recognize a specific type of structure. And T cells recognize the peptide offered by MHC component. Very different. And B cell receptor can also offer diversity, specificity, and recognize self and non-self. So why we start first? So we start with T cells, and actually there are so many common things between T and B cell, and we will mention later. So what is the T cell receptor look like? And for CD4 T cells, they have alpha chain and beta chain. And for CD8 T cells, they look quite different. They have a one longer chain and one smaller chains, not shown here. But anyway, and the MHC component for class 1, they look like this. It look like a sandwich, like a clamp. And class 2 also looks similar. But class 2 allow the longer peptide because they have a longer edge. And the component also look like, oh, sorry, I'm wrong. MHC class 1 look like, I think, I'm sorry. I was wrong. What I mentioned for T cell is actually MHC class 1. Sorry, it speaks too long, too fast. But anyway, MHC class 1 can offer a shorter peptide for CD8 T cell, and MHC class 2 can offer longer peptide for CD4 T cell because the edge here is not tightly closed in comparison to class 1. So usually the peptide presented by MHC class 1 is 8 to 10, and the peptide presented by MHC class 2 can between 13 to 17. And just imagine our amino acid have 20 kind of amino acid. So within this 8 to 10 amino acid, actually can have different kind of composition. So actually, and this peptide presented by MHC molecule can offer different kind of composition. And here is the analyze to see whether the peptide offered by class 1 or class 2 are really randomly can be composite by 20 different kind of amino acid randomly. But actually, it's not true. Actually, have some anchor residues that cannot be changed randomly. And here is how the CD4 and CD8 as co-stimulator look like. For CD8, they specifically recognize class 1, and the CD4 is specifically recognized class 2. So what is MHC class 1 and class 2? As we know, class 1 and class 2 have to present peptide to CD8 or CD4 positive T cells. So it will be very important which kind of cells can offer class 1 molecule or class 2 molecule as the so-called antigen presenting cells. And this table gives you a summary which kind of cells present class 1 and which kind of cells present class 2. So anyway, for all the cells that can present MHC class 1, they have chance to elicit CD8 T cells response. You can see that nearly all the cells besides red blood cells can present MHC class 1. On the other hand, MHC class 2 is quite specifically to the specialized antigen presenting cells, such like B lymphocytes, macrophage, dendritic cells, and some epithelial cells, especially in GI tract, mucosal tissue. Let's continue. So in overall, which kind of peptide could MHC class 1 antigen presenting cell or MHC class 2 antigen presenting cell be presented? Most of the peptide presented by MHC class 1 came from the endogenous protein, means this protein need to be generated in the antigen presenting cells. And for the peptide offered by MHC class 2, they usually acquire from exogenous antigen. They firstly eat from outside and get inside, digest cells, and therefore present outside. That is the major difference between the class 1 type peptide and class 2 type peptide. And how the peptide offer belong to MHC class 1. And as we know, the protein translation happens inside the cells, right, through ribosome. And according to the textbook, it seems that there are huge proportion of the new synthesized protein will be delivered through the TAP1 and TAP2 protein. And that allow the component, this complicated component, and that compensate the premature complex of MHC class 1 molecules, and therefore digest it and load the peptide on the surface of the class 1 molecule, and therefore be delivered to the cell surface. So according to this theory, the peptide that can be only presented by MHC class 1 need to be generated inside our cells. And the one pathogens that definitely do that is virus, because virus generate the viral protein inside our cells. So class 1 will be very good for killing the virus and also the abnormal antigen, mutated antigen for the cancer cells. So CD8 T cells is very important for curing that. And for class 2, they will be mostly came from the exogenous antigen, firstly by endocytosis and go to early endosome, later endosome, and therefore lysosome. And this component will therefore digest the protein into small pieces. And on the other hand, the endoplasmic reticulum also secrete the premature MHC class 2 complex. And when they go to the acid environment, they will also be separated, and therefore allow them to load the digest the foreign antigen peptide from exogenous environment, load onto the MHC class 2 molecule, and therefore to present it on the surface. Yeah. But it is not, although it is a general idea between MHC class 1 and class 2 peptide, but actually there are some reaction also happen called cross presentation. Means the peptide that should be delivered into MHC class 1 may also have chance be delivered into MHC class 2. For example, autophagosons, they digest the component inside the cells into small pieces into the vesicle, and therefore these chains will allow the MHC class 2 to load these the antigens that actually generate inside the cells into they recognize it as a exogenous antigen. And also some cases, the class 2 molecule peptide can also be delivered to MHC class 1. It calls cross presentation. And it might be quite important for the eradication of some viral pathogens. And the other thing of the MHC molecule is this molecule itself actually is highly diversified in between every single person. Firstly, our body actually creates one set of MHC class 1 and MHC class 2 from our mother and our father. So we have different pairs from mother and father. And because their sequences are quite similar, so during the germline, they might have something called recombination and change into a nearly new version. So theoretically, unless in human beings, unless these two peoples are identical twins, or their MHC molecule must be different. So you can consider MHC molecule is the antigens that different from every single person. And it also become the main reason of the alloreaction, means the tissue rejection when you have tissue transplantation. Actually, the main reason why my body rejects the organ from you, perhaps, is mostly due to my body's T cells attacked the MHC molecules presented by your cells, because it is the most diversified protein in between human population. And of course, due to this diversity, it also allowed different individuals present different portfolio of peptide sequence. Understand? For example, perhaps my perhaps the genetic of my MHC molecule not allow me to present an antigen specific from a specific type of virus, but maybe you can. Maybe Lisa can. Yeah. So after the outbreak of the virus, I will die and you too will survive. It's also a way to help people, help a population of organisms not die due to a specific type of pathogens infection. I cannot protect. You can. Good. Oh, there's so many. So I think we have already mentioned antigen-presenting cells and MHC class 1 and class 2. And we also mentioned what is antigen. Antigen actually is a short peptide recognized by T cells, and for structure recognized by B cells. And therefore, we need to talk about what causes the immune diversity in our adaptive immune response. I think most of you should learn this table before, no matter you learn immunology or not, because this table should be present in the textbook of molecular biology. But anyway, the component of the T cell receptor or B cell receptor, especially the hypervariable region, means the place they recognize antigens, no matter T cell peptide antigen or the B cell antigen as antibody. And actually, each BCR and TCR, they are generated by the recombination of different components like TCR alpha chain here or beta chain here. They actually compound with many different kind of V variant, J variant, and by random recombination, they can generate into different kind of composition for the alpha chain and beta chain. And the difference between alpha chain, beta chains can therefore offer more different kind of T cell receptor inside every single T cells. And also B cell also have the same components in their germline DNA, genomic DNA. But if they want to be composite into a functional T cell or TCR or BCR, they need to do recombination and select each component only one. And this mechanism is quite complicated. I think I will not mention today. But not only the recombination and this protein that allow the this VDJC component combine into a new B cell or T cell receptor. They can also digest and therefore allow the non-homologous end joining that get more diversity into the junction region. So anyway, there are many different kinds of T cell and B cell and T cell receptor, especially the hypervariable region. And you can see that according to the different component of V variable segment, D segment, or J segment, blah, 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 you can fold them, and ultimately, and in T cell alpha chain, beta chain receptor, when they come together, they can offer up to 10 times to 18-fold of diversity. And for B cells, minimal can offer 10 times 14 kinds of B cell receptor. And B cell have another thing called affinity maturation that can even offer more kinds of B cell receptor thereafter. So anyway, just believe this number is already very crazy. That should be allow our immune system to recognize any single kind of foreign antigens we might encounter. Do you believe? You believe? You should believe. Yeah. So anyway, it's good. They offer such kind of diversity. Is good, very good. But this diversity might also include our own antigen, right? Our body antigen, like actin, beta actin, GAPDH, whatever. Yeah. So this diversity might also acquire our self antigen because our self antigen can also be presented by MHCs class 1 and class 2. And that should also have chance to stimulate T cells or B cell immune response. And I think I remember Professor Huang told me I need to have a 10 minutes break at the middle time. So now it's 11:05. So we take 10 minutes break. We come back after quarter past 11. 美女，这个是要继续给他录吗？还是可以暂停啊？嗯，又得上完一半了。 现在是在线上。啊，喂喂，你好。Yeah. Oh, so we continue. So we finish. I think somebody asked some question. I think I will reply in the later part. Somebody asked TB and inflammation. I think in our slide we'll mention that later. So anyway, we finished the introduction how the adaptive immunity offer the possibility of diversity. And this number is a lot. I think diversity should be enough. But another issue is how the immune system, after getting such amount of diversity, can still not recognize self-antigen because this diversity is totally random. So it offers the chance this diversity can also recognize our antigen present our self peptides. Then it will cause autoimmunity. That will be very bad. So before we talk about how the adaptive immunity recognize self and non-cells, before that, we need to mention what is co-stimulation molecule. We call this B7.1, CD80, CD86, and CD28. And I need to mention to you the functional activation of T cells require these three signals together, or they will not be fully activated. So here's a very complicated. Oh, this thing appear again. Anyway, it is a very complicated figure offered by Nature Immunology, as I say, immunology like to do that. But anyway, they put all the so-called positive co-stimulation marker on antigen-presenting cells or T cell, or the negative co-stimulation marker on antigen-presenting cells or T cells in this figure. Looks very, very complicated, but just let you understand the T cell immunity, whether the T cell after meeting antigen, either these T cells is going to be activated or going to be silenced. It depends on which co-stimulation marker be offered from antigen presenting cells. Just have this idea because there are too many, you cannot remember. Some of them are positive signal, some of them are negative signal. And also, even on T cells, there are some offer positive signal, some offer negative signal. Too many combination. This thing never disappear. Oh, they disappear. And here is just an example how people utilize this so-called positive signal and negative signal of co-stimulation. The most famous one is the antibody targeting CTLA4, and the other one is the antibody targeting PD-1 and PD-L1. I think you might know that it's a very hard immunotherapy strategy people are using now. According to the literature, this antibody might cure 50% of human cancer. So why human cancer? Why this antibody, after blocking the interaction between PD-1 and PD-L1 interaction between APC and T cells, they can cure cancer? Because cancer cells present lots of PD-1 and PD-L1, and they can't let the nearby T cells going into silence and not attack the tumor cells. So this blockage can remove the inhibitory effect of the PD-L1 and PD-1 reaction and reactivate T cells. And the other antibody targeting CTLA4 also have similar effect, but just not as good as the targeting PD-1 and PD-L1. So the thing I want to tell you is which which co-stimulation molecule present by antigen presenting cells actually depends on the cell type and also the environment or the history of this antigen presenting cell be exposed. For example, the dangerous signal offered by toll-like receptor allow this antigen presenting cell present these two molecules called CD18 and CD86 at high level. And these are very two strong activated signal. And after combining to CD28 of T cells, and therefore T cell will be highly activated. Okay. So when the T cells, if their TCR recognize the peptide presented by either MHC class 1 and class 2 without the proper positive activating costimulation signal, and this T cell will have three fates. One called clonal deletion, means they will go to apoptosis, die. And the other one called energize. Energized states means these T cells, their chromatin are changed foreverly by epigenetic modification. And these T cells, they will not die. They can still be activated, but they need extremely strong signal to be reactivated. Okay? And the other one is this T cell might become a specific type of T cells called Treg, regulatory T cells. And these regulatory T cells can inhibit the overimmunity of T cell response. So this is very important because this knowledge help us to understand why these diversified TCR and BCR not recognize the self-antigen. Because this theory not only happen in T cells and also in B cell. B cell also have their own costimulation marker. So for B cell development, they happen in bone marrow. And the bone marrow offer interleukin 7, and that support the development and survival of B lymphocytes. And they will go through many different stages and eventually become naive B cells, and they have a surface IgM on the surface. And this one is the very detailed development, every single step of B lymphocytes in as long as their signal pathway. And it is the gift for you. You can read that after. It's too complicated. So anyway, when the T cells get development, especially the generation of the initial B cell receptor on its surface, we need to understand our body actually are mostly in steroid situation, right? So means if the B cell after development, when they meet our self-antigen, actually is in the situation without dangerous signal. Quite easy to understand. So quickly after development, if the B cells have chance to recognize self-antigen, or no matter in large scale or low scale, they can switch to either clonal deletion, apoptosis, or go to energize. Quite can understand? So for T cells is a little bit more complicated. For T cells, their progenitor derived from bone marrow cells, but for T cell development, they need a transcription factor called FOXN1. But anyway, after the cell acquire this one, they will go to an organ called thymus. And this thymus and T cell development happen very, very activated before our teenager age. But after we get adult, and this T cell development becomes slower, and eventually quiescence. And in the old man or the aged people, their thymus will turn into the fat tissue. So anyway, the T cell diversity usually happening during our young age. And for T cells, especially the thymus, historically, they can be separated into two parts, one called cortex, and the other one called medulla. The cortex epithelial cells, they have chance to offer the MHC class 1 and MHC molecules on their surface. And And also the medulla, they also have the MHC class 1, class 2 presented by its surface. And one thing very importantly during the T cell development, they have 4 stages called double negative 1, double negative 2, double negative 4, and single positive. And therefore, but the most important thing I want to say that during the cortex, their life in the cortex, the requirement for the survival is its T cells need to have the ability to recognize the MHC molecules present by the cortex epithelial cells. If they cannot attach or have no affinity, no matter at this stage, no matter the affinity is strong or weak, fine, they will survive. But if they cannot attach, these T cells will die. And this consequence is the combination of many different signal pathway, and then that make this fate. So the most important thing during cortex development is the requirement of the TCR have ability to recognize MHC molecule. So that can cause something called T cell restriction. And this selection we call positive selection means the T cells that can bind to our own body's MHC molecule, these T cells survive. And therefore, when they move to medulla, and this medulla epithelial cells is a very interesting kind of cells because they have 2 spatial transcription factor. One called AIRE, a very famous one, and the other one called FEZF2. AIRE is the most important one. And this transcription factor allows the cells randomly transcribe the cellular RNA inside the cells. We have to know that our body have many cells, but every single cell looks differently because every single cell expresses different RNA. They have the same genome, but they express different RNA. So that's why we have liver, we have gut, we have lung, everything looks different because they express different RNA. And these medulla epithelial cells is quite interesting. Due to this AIRE transcription factor, they are allowed to randomly transcribe the cellular RNA from the different genome part. Means they might express the antigen from pancreas, from gut, from brain, from anywhere. So that allows the medulla epithelial cells express any single self antigen inside our body, and that can recognize by either CD4 or CD8 T cells. So at this stage, because the lackness of co-stimulation marker, so when T cells recognize the self antigen very strongly, they will die. So this stage called negative selection. So ultimately, the T cells in our body or the diversity of our body, they exclude the T cell that equipped with TCR that recognize our self antigen. Of course, partial of the T cell recognized self antigen will be generated into a special cells called regulatory T cells, but it will not be mentioned in today's lecture. It's too complicated. But of course, they still have a small chance that part of the T cells, they hadn't meet the self antigen, but go to the peripheral. So they are still self-reactive T cells. But thank God, because our peripheral tissue theoretically should be in steroid position, environment. So it means even they go to peripheral, it's still lack of so-called dangerous signal. So they still have a chance to give tolerance outside the thymus. So it's called peripheral tolerance. But of course, if 2 mechanisms both failure, then ultimately this individual will get autoimmunity. So it's quite, so sometimes actually there are many literatures mention that autoimmunity usually happen after a very bad injury or inflammation. I think you can understand why, because perhaps it's the reason the severe, should not say severe, very badly wound or the infection that make this peripheral tolerance failure. Because they offer the dangerous signal. So anyway, I already mentioned the diversity specificity of T cells. So ultimately, after this so-called selection, our T cells and B cells should exclude out the possibility of recognizing our self-antigen. So ultimately, the T cell that survive can at least bind to MHC weakly. That called MHC restriction. Your T cell at least need to have ability to bind to T cell to offer the survival signal. This signal is offered by selected by positive selection. And the T cells that recognize the self-antigen peptide complex very strongly either die or energize or become T regulatory T cells. And sometimes people might consider why our body need to keep the self-reactive TCR or BCR cells inside our body that can recognize our own cells into energized stage, but not just simply let these cells into apoptosis. One explanation is perhaps our body want to keep these T cells or B cell repertoire in case we meet a very deadly pathogens and their peptide or antigen very similar to our own antigen. I think this explanation quite makes sense. We keep these cell-reactive T cells or B cells in energized stage, but if we want to activate that, we need very, very, very strong stimulation. So it should be still fine. Yeah. So after I explained quite many components of immune system, so we can go back to explain what is NK cell. NK cell is a cell that have many granular, toxic granular that can kill the cells that recognize. And what NK cells recognize, it is not as diversified as T cells or B cells, because this NK cell recognize two categories of cells that are considered as abnormal cells. One is the cells expressed with extremely low level of MHC class 1. MHC class 1 should be present by every single cells inside our body. And it is important to help us exclude out the possibility of the cells that infected by the virus or the tumor cells. So it is also a strategy how the virus or the tumor cells escape from immunity by the reduction of MHC class 1. So NK cell now will become very important. NK cells also have the so-called positive signal and negative signal, and their corresponding receptor. For example, MHC class 1 offer the negative signal for NK cell activation. So because normal cells usually express MHC class 1 ligand at normal or even high level, so it will not cause NK cells become angry and kill that cells. But when the tumor cells or the viral infected cells, they tend to reduce the expression of MHC class 1, and therefore NK cells can target these kind of cells, even without the presenting of foreign antigen. So they recognize the abnormal cell. And the other thing is the cells that are very stressful, very tired, very stressful, such like they meet some problem for metabolic or some problem for blah, 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 many other things. Yeah. So NK cells therapy actually is also quite popular recently. Some company actually offer the adaptive transfer of the NK cell derived from yourself and expand outside and therefore inject back to treat, to make you healthy. and it works quite well for certain type of cancer. But remember, these cells even not offer diversity, but they can do that. They recognize the stressful cells or the abnormal cells. Oh, how to make this thing disappear. So after that, the next question is, okay, we talk about adaptive immunity and how they are activated, how they offer diversity, specificity, and how they exclude our self-antigen. So now the question is how they meet antigen. In the other word, how antigen-presenting cells offer antigen to T cells. Actually, this thing happens in the lymphoid organ, and more specifically, lymph node or the lymphoid tissue or spleen. And our body, beside the blood vessel, we also have the lymphoid vessels, and that can circulate this body fluid in the lymphoid tissue. And these lymph nodes are very important filtrate device that can allow the filtration of the foreign antigen to this lymph node, and also the recruitment of the activated antigen representing cells homing back to the lymphoid organ. And here is how the a lymph node look like. And you can see that the lymphoid fluid will be go inside from here and therefore be go out from here. And they also have the blood vessel can allow the circulation of the immune cells when they are going into blood vessel go back to the lymphoid. And the entering of the place that allow the immune cells go back to the lymphoid tissue is the full name is high endothelial venules, HEVS. And you can see that the substructure of lymphoid tissue, they are very specifically, the blue part is the zone called T cell zone, and they usually are aligned with the blood vessel. And the outer sides are the B cell zone, and this bright yellow part are the activated B cell zone. And we will mention that later in humoral response. It means the antibody response, how antibody be activated later. And the spleen is also have quite similar structure, but not as organized as lymph node. They also T cell zone align with the blood vessel and the outside B cell zone. And not even the lymph node and the spleen. And in the other place, like our gut system, we have also have the lymph node-like structure called Peyer's patch or mesenchymal lymph node. And for Peyer's patch, the out layer, these red cells are the gut epithelial cells, and beneath that you can see that is the T cell zone, and also the outer layer is a B cell zone. And these M cells is have a similar function like the HEV that deliver stuff from blood vessel into or lymphoid organ into lymphoid fluid into the this sublymphoid tissue. So therefore, how the antigen be delivered through by antigen presenting cell to this lymphoid tissue. And firstly, we need to mention 3 different kinds of antigen presenting cells. One called dendritic cells, and they have many legs, many dendrites. That's why they call dendritic cells. And they are everywhere, and they like to eat. And the other thing is macrophage, and they are mostly reside beneath our skin and also in many other tissues. And the other thing is B lymphocytes. And B lymphocytes, the cells specific for making antibody. So actually, the key player for adaptive immunity is dendritic cells. They continuously eat something from outside into their own body and present to outside. And here is one very interesting photo offered by the Janeway immunology, you can see that the dendritic cells, when they are in the peripheral tissue, and they are two color. The green color is ANC class 2 because they eat something from outside to inside, and the red color is lysozyme protein. You can see that when they are in peripheral tissue, they already express high level ANC class 2 and continuously digest something from outside to inside. But when they are activated, when they are transferred from the peripheral tissue in the way to the central lymphoid tissue, they change their shape and start to digest the eaten peptide and present to MBC class 2. And when they go to the lymphoid tissue, they are fully activated and become the fully functional antigen-presenting cells. And dendritic cells is the most important antigen-presenting cells that transfer the foreign antigens from peripheral to central immune system. But also macrophage and B cell can also do that, but their function is more like the booster effect, but not the primary effect. And there are many ways the dendritic cells can eat something from outside to inside. And here's a more detailed photo showing how many different kind of receptor and co-stimulation marker on a dendritic cell, specifically the activated dendritic cells. So they are very strong antigen-presenting cells after engulfing the antigen, specifically after they engulfing the dangerous signal. And after that, they will go to the lymphoid tissue and therefore activate the T cells that have the T cell receptor corresponding to the MHC molecule and the peptide complex. And the more detailed information of the macrophage and the B cells are described here. And they have a more strong ability to boost the immune response. But as I said, the most important primary boosting antigen-presenting cell is still dendritic cells. So again, there are many companies offer some therapy called dendritic cell therapy. So what they perform, they firstly isolate your blood cells and try to generate or isolate some dendritic cells and give them some dangerous signal, or make them, for example, let them exposed to the tumor section acquired from your body, and make them eat then foreign antigen plus dangerous signal, and therefore inject back, and you will have a good response. This thing already have many clinical data. So there are many, and here is an overview of 3 different kind of antigen-presenting cells. And as I said, dendritic cell is the best one. They can give the strongest signal, and B cell and macrophage more likely to become the booster effect. And after the antigen-presenting cell offer the correct MHC molecule plus peptide to TCR, and therefore the T cell, the alpha-1 antigens were locked to the ICANN offered by the antigen-presenting cells and be strongly affront each other. And therefore, the T cells activation eventually happened after we talk so many things. And activate T cells will further proliferate through the activation of different cytokines, especially the interleukin-2 is a very strong T cell proliferation cytokines, and therefore have a clonal expansion. Clonal expansion means a T cell with a specific type of TCR be expanded to recognize, to have in order to perform their function. So what happened after the activation of T cells? Again, their surface molecules will change. And please take a look of this table very carefully. Resting T cells means the T cells are still in naive stage, means they hadn't been activated. They will express the different component of surface marker. They will allow them to be re-entered back to lymphoid tissues to let them have chance to meet the foreign antigen presented by antigen-presenting cells. And this molecule help them to recycle back to central lymphoid tissue. But after they get activation, you can see that this molecule disappear. So they will be enforced go out to peripheral. And if you still remember this thing, if I remember, there's one slide I showed Donald Trump, location, location, location. And this is exactly the interchange that help this activate T cells stuck to the damaged place. If you remember. You remember? Remember? You don't remember? I will have exam. So after the T cell activation, if you still remember, the activation of T cell requires 3 signal, MHC peptide and TCR and cosmation marker, and the third one is cytokine. And this cytokine will decide which kind of CD4 T cells become. Yeah. For example, if they meet different kind of, sorry, I should mention this one first. During the time when they get activation, if they meet TGF beta cytokine, they will turn to Treg cells, and by the activation of the corresponding transcription factor. And if they meet interleukin-6, they will become T follicle cells, T follicle helper cells. If they meet the combination of these two cytokines, they will become T helper 17. If they meet the cytokine that tend to have inflammation, they will go to T helper 1. If they meet the IL-4 cytokine, the transcription factor GATA3 will be activated, they will become T helper 2. So this is a third signal offered during T cell activation. So of course, these T cells have different function, and that will control the direction how our whole body immune system should move forward. What is? It sounds like toilet. No, whatever. So for the most widely mentioned T helper cells are T helper 1 and T helper 2, because T helper 1 stimulate inflammation response, and they can further activate the macrophage become even more crazy stage. So I remember there's one student asked how the inflammation contribute to the clearance of TB bacteria, and that is the explanation. The cleaning of the TB bacteria intracellularly in macrophage require the help of T helper 1 T cell immune response. So it also although this is a bacteria, but the cleaning of this TB infection require adaptive immunity. And the T helper 2, they will secrete many cytokines that facilitate antibody response. It's a very strong growth factor for B cells. And T helper 17, T follicular cells, and Treg cells also have their different function in regulating immune system. And one thing very importantly is CD4 T cell is the central of not only adaptive immunity, but also innate immunity inside our human body. For example, by using the genetic knockout mouse experiment, it already validated 20 years ago, if a cells without CD4 positive T cell, the CD8 T cells inside these mice also have no function or not have fully function. It cannot kill the target cell efficiently. And here is just the explanation to tell you the interplay between how CD4 T cell help the function of CD8 T cells. It's so thirsty. And here is the slide showing you how the adaptive immunity further go back to facilitate or activate the innate immunity. For example, they can further secrete, for T helper 1 T cells, they can secrete many different lymphoid, lymphocyte growing factor, or many other chemokines or inflammatory cytokine that further stimulate the activation of macrophage and allow these macrophage to digest the intracellular pathogens in more efficient way. So if you want to kill the intracellular pathogens, bacteria in macrophage, you really wish your immune system switch to T helper 1 like immunity, because mostly our immune system, if you categorize into very briefly way, your immune system after the activation either will switch to T helper 1 like immunity or T helper 2 like immunity. T helper 1 like immunity more tend to elite cell immunity response, means the activation of CD8 or the activation of macrophage and other innate immunity cells to make them crazy, and T helper 2 more tend to elite the humoral immune response, means the secretion of the T helper 2 type antibody, and I offer the neutralization antibody and also activate the basophil and eosinophil that combat the parasite infection. So if you understand these two ways quite important. So if you really want to cure the intracellular bacteria, you better wish the immune system switch to T helper 1. So what will happen if the immune system switch to the wrong way? And for example, the TB infection. TB infection is an intracellular pathogen. So after the macrophage digest, engulf this kind of pathogen intracellularly, they will not be able to degrade them instantly. They need the requirement of the activation by T helper 1 CD4 positive T cells. But if unfortunately these individual or patients, their immune system tend to T helper 2, and therefore these T cells will surround the infected tissue or infected macrophage and try not to allow them to go out or spread out, and they will form a spatial structure historically called tuberculosis. So it is how the T helper cell decide the fate of the consequence of immunity. This page. So adaptive immunity can further make further boost innate immunity. The best example is macrophage. They will from here to here and become totally crazy. And here are the many cytokines and their effect on the different type of lymphoid tissue and the macrophage and the consequence. And just tell you that different T helper cell will secrete different kind of cytokine, and they will have different consequences. So as I say, immune system is not perfect. We anyway will get disease, but we need to know how to fine tune our immune system into homeostasis and help us to cure the disease that is ongoing. So knowing these T helper cell and cytokines can allow us to treat the disease and how to say fine tune our immune system in the correct way. And this cytokine theoretically we can also synthesize in the laboratory or in the industry. So we mentioned so many things about CD4 positive T cells. So how about CD8 positive T cells? CD8 positive T cell is then called cytotoxic T cells. So it should be quite easy to understand after this T cell recognize the antigen presenting cells through MHC class 1. You need to understand every single cells have MHC class 1. So theoretically, it means it have ability to kill any single cells that express the foreign antigen with the proper costimulation marker. And it can kill these cells by many different ways. One is by fast ligand-induced apoptosis, or just deliver many toxin stuff from the CD8 positive T cells. They inject into the cells that targeted. And there are many enzymes that can either make the targeted cells have pause For mammalian cells, if they have pore, it definitely die, and many enzymes that can cut the protein inside the cells and finally lead to apoptosis. So it's a very efficient way that CD8 T cell kill the target cells. And after T cell proliferate be activated, they will go into a highly expansion way called clonal expansion, and few of these cells, they will change their surface marker as well as the transcription factor to allow them have the memory effect. So of course, after the T cell, after the clearance of pathogens or so-called foreign antigens, these clonal expand T cells will eventually die due to apoptosis. But few of them, during the expansion, if they acquired the activation of these transcription factor and also the equipment of these surface molecule, they will allow these kinds of cells to survive, especially by the highly expression of the interleukin-7 receptor. It is a very strong lymphocyte survival factor, and also the very strong expression of BCL2 is a very strong protein for anti-apoptosis. And therefore, these T cells will allow to survive as memory T cells and keep inside our body. In the future part, if we meet the same antigen, they will be activated very quickly in the next time. So that's why vaccine work. 我们什么时候结束啊？12点半啊？好吧。So let's 12点半吧。So after we talk about so many T cells, let's go to talk something about B cells. So just remember, B cells recognize structure. And this is how the antibody look like according to their antibody, according to the amino acid composition or the genome composition. You can see that, and the x-axis is the location and y-axis is the variability. You can see that the antibody, no matter the heavy chain or light chain, and the hypervariable region are mostly located in certain region. And these regions are usually composite the antibody at the antibody recognition site of an antibody. And how antibody recognize the so-called antigens, as I say, by structure, but not by covalent bond. They use the such like electrostatic force, hydrogen bond, or van der Waals force, and hydrophobic force. And by doing this force, they allowed, in combination of the high diversity of B cell receptor, theoretically, the B cell repertoire inside our body should be allowed to recognize any single kind of foreign antigen. 到什么时候？哦，到12点半。Yeah. So we have already mentioned how the B cell diversity have been generated. And we only mentioned the hypervariable region, VDJ recombination. But we hadn't mentioned what is C. C means constant region. And the constant region of antibody is specifically important because it will be important for something called the antibody class switch. For example, IG, IA, IgN, IgG, IgE class switch. But just this figure just led you to understand the difference between the B cell receptor and secreted immunoglobulin. They are actually derived from the same gene, but they use different kind of RNA splicing. For the membrane bound one, they will keep the cytoplasmic domain, so that allows them to dock on the surface of the cells. On the other hand, for the secreting antibody, and they will have the early splicing part before the cytoplasmic domain, and allow them to be secreted outside the cells as the antibody. And some antibody isotype have a dimer or the pentramer, like IgA is formed as a dimer and IgN is as a pentramer. That learn to recognize more antigen as a complex. So again, T cell, similar as T cell immunity, B cells activation also require different signal. First one, of course, like T cell receptor and MHC class 1 antigen, their BCR need to recognize the antigen. And the second, they need the co-stimulation marker offered by T helper cells. Surprising? Because this co-stimulation marker is called CD40 ligand. It is offered by helper T cells. And the co-receptor, the corresponding receptor is CD40 on B cells. And this activation also requires the T helper cell to secrete the corresponding cytokines like interleukin 4, interleukin 5, interleukin 10 are very good for B cell proliferation. So they also require 3 signals like T cell activation. But of course, in our immunity, our body keeps some B cell repertoire that don't require so many complicating T cell helping. And this kind of B cell, once they recognize the antigen, especially the spatial structure from bacteria have PAMs, they can be directly activated, called the thymus independent antigen. How many slides I have? So what antibody need to do when the B cell become activated, then they will be proliferating and eventually become a stage called plasma cells. At this stage, they will crazily secrete antibody outside. And what antibody can do, the first mechanism is they can neutralize the pathogens. And this one will be extremely important for the virus infection, because if our body have the antibody that recognize the viral glycoprotein required for cellular binding, and therefore this neutralizing antibody theoretically should allow our body prevent from this virus infection. And the other thing is opsonization, and because there are many the surface protein called Fc receptor that will be created by many immune cells. And this Fc receptor can recognize different kind of antibody, and that can stimulate the engulfment of the cells that bound to the antibody complex through Fc receptor. And of course, as we mentioned at the very beginning, and antibody can also stimulate complement activation, especially IgM. Oh, no, this one is very complicated. And let me see how many slides. Oh, 7 slides. And here, this photo just want to tell you how T helper cells help the activation of B cells. You might consider it sounds quite strange because B cells recognize structure or the protein structure. But T cells recognize peptide. So why B cell activation require T cells help? I think it's, and the explanation might be, you can easily see, if you remember the lymphoid structure, around the blood vessel is the T cell zone, and thereafter is B cell zone, the yellow part. And the in-between region is exactly the place where B cells and T cells can meet each other. So actually, it's quite easy to understand. If a B cells recognize a structure that can be considered as a foreign antigen, so it can also stimulate T cells. So if T cell is also an antigen presenting cells. So when the BCR bind to the antigen, they will cause endocytosis of antibody antigen complex. and therefore be presented to the surface through MHC class 2. So it is by the requirement of T cells activation means this antigen can further be recognized by T cells through peptide presented by B cells. And these T cells can therefore offer the signal of the B cell for Foley activation. I think it's a further step for controlling the foreign antigen during the evolution. I think quite makes sense. And where the antigen get into lymph node, and you can clearly see that according to this figure, and these antigens can be delivered from our body fluid, go to our lymphoid tissues, and therefore go into the lymph node from the afferent lymphoid vessels. And therefore, there are many macrophages beneath the outside layer of the lymphoid organ, and that have chance to trap this antigen, and therefore transfer this antigen to the follicle dendritic cells, and that can therefore be recognized by B cells inside our lymph node. Very complicated. And after the B cell activation, again, their surface marker will change. And for the naive B cells, they have extremely high level of surface antigen, and also extremely high level of MHC class 2, because they are both required for the antigen recognition on the active and the recognition of the corresponding T cells. And there's no antibody secretion at all. But when they meet antigen plus the stimulation of T cells, they will change to the plasma blood cells at the crazy growing stage, and they start to produce antibody. And therefore, when they are fully activated, after a stage called somatic hypermutation, or the antigen or affinity maturation, I will mention later, they will become a fully function activated plasma cells, and they do not, or they express extremely low level of surface antigen and MHC class 2, because they don't require it anymore. And therefore, they will secrete antibody at extremely high level. And here is the requirement for antigen stimulation, because they are already fully functional, so they don't require. And this one is a very complicated stage called somatic hypermutation. Actually, when the B cells meet antigen, this B cell not necessarily, the BCR of this B cell not necessarily have to bind to this antigen at extremely high affinity. It can bind, it can stimulate, it can be activated, but maybe it is not perfect. So here's one stage happening in the germinal center of lymphoid tissue. And this germinal center have a whatever light zone and a dark zone. And during this time, you can see that this germinal center is surrounded by T lymphocytes. So anyway, here's a precisely proliferating T cell, B lymphocytes, and they will meet, and at this stage they will meet, and they will have a very spatial stage called affinity maturation that allowed the B cell, or the variable region of B cells, to get further diversity, to let them have chance to increase the affinity that bind to the corresponding antigen. And only the one, or so-called a clone, when the B cells get mutation, they might have different, by this enzyme called AID, and they can deaminate, there is an enzyme called UCL DNA glycolase, and they can remove the pyrimidine residue from the nucleotide, and therefore make damage and cause randomly fixation. And this can allow the B cell diversified region further get more diversity. Understand? And this diversity might have different consequences. For example, after fixation, if this fixation is not in frame, means these B cells cannot express the functional B cell receptor, and this cell definitely die. Okay, And the other thing is the new generation B cells have no chance to bind to antigen, they die. And by a very low chance, these B cells, after getting so-called somatic hypermutation or affinity maturation, they will have chance to generate perhaps one single cells that have extremely high affinity binding to the antigen. And these B cells will therefore be further clonal expanded, become the major population of the B cells that secrete the antibody targeting the antigen. So actually, there's one thing where, although we have many different ways that can screen the so-called artificial antibody by fudge display or different way to generate the antibody we want, but only inside the human body allow us to get the best antibody through this way called affinity maturation. 知道12点半吗？ I think it's the final 3 page. So we have already mentioned the diversity of B cell receptor, right? So now we talk about their constant region. So their constant region actually have many options. One called mu chain, delta chain, gamma 3, gamma 1, gamma 2b, gamma 2a, epsilon, and alpha chain. And what calls antibody class switch. And I remember there's a cytokine. So it depends on when B cells get activation, the surrounding T cells offer which kind of cytokines to this kind of B cells. And they will change to different, they will choose to have different kind of constant region and therefore generate into different kind of antibody class subtype. And the different subtype will have different function. For example, some subtype like IgG1 in human body, they have a strong ability to have ozonation. And IgM have the best for the complement activation. IgG3 also have very good complement activation. IgA is good for neutralization, especially for mucosal system. And IgE is very good for stimulate mast cells. So it depends on which kind of cytokine he encountered during activation, and they will decide to switch into different kind of antibody subtype. And it is their function, and their distribution will be also different. IgA mostly in mucosal, and some antibody class can transport out, go across placenta, can get protection for the baby, and blah 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 blah. Very complicated. And I think here is the final stage that show you the location of different kind of antibody subtype, and you can guess what is their function, what they mainly protect from. And finally, the information I want to tell you is that even the small toxin or very small chemical can also be allowed to be recognized by antibody if you have a proper strategy. Yeah. So I think we still have 15 minutes. I know you have exam, so maybe I can give you some hints. So let's have some game. So if you have wounds, most of the bacteria are killed by which one? If you want to leave earlier, answer which one? A, B, C. A, correct. Yeah. But if you go worse, you still need adaptive immunity. So if you get one, most of the bacteria are killed by which cells of innate immunity? Excellent. So if you get wound, most of the bacteria are killed by which non-cells of immunity? Yeah. It will be multiple choice. And if you have a really bad bacteria infection, especially the intracellular bacteria, which type of T helper cell you need? And I know you are hungry. T helper 1. Which immune cell play the key role for killing virus infected cells? But the main part is still CD8 T cells because they anyway recognize foreign antigen. Oh, so the more complicated one. So there are many vaccines for COVID-19 now. So the COVID-19 is a syndrome, actually recognize SARS-CoV-2 virus. So there are Moderna, and it's a mRNA vaccine. AZ is a adenovirus vaccine. And there's another protein vaccine made in Taiwan, and the other one vaccine is made in mainland China. It's a killed virus vaccine. Which one can cause CD8 T cell response? Huh? mRNA, yes, and huh? AZ. Because CD8 T cell recognize class 1. Class 1 antigen mostly come from the endogenous protein. Means the protein need to be generated from antigen-presenting cells. So only mRNA produce protein, and virus infection, they produce viral protein inside the cells. And this one, I cannot say cross activation will not happen, but if answer should be these two, because I say highly possibly. Highly possible, sorry. Should be no two. So which of them suppose can create neutralizing antibody? Huh? What? No, all of them. Because they all pass clinical trial, come on. So I remove the company name. Which one theoretically might have the worst antibody affinity for the virus? Last one? Correct. Because antibody recognize shape. If you kill the virus, no matter by heat or by chemical, you nevertheless might change the shape of the virus. Understand? What else? You get side effect of the vaccine like high fever, mostly due to adjuvant. So do you know what is adjuvant? You remember the activation of immune response need co-stimulation marker. So this adjuvant actually offer dangerous signal. And this dangerous signal, that is the reason make you feel unhappy. And what is the question mark? Autoimmunity, yeah. Which antibody is supposed to be the best one that protect you from respiratory virus? Huh? No. Can? I mean the best, suppose. No. IgA, because it's in mucosa. Why anti-PD-1 antibody cure cancer? Because it block the negative signal of T cell activation. If this one interact to this one, then T cell get silenced. And this antibody, and some company of antibody here, they just block the binding between these two. Tumor cells usually offer PD-L1, and T cells offer PD-1. Final one. So if you want to block this one block negative, how to say, this one stimulate T cell response. So if you want to block hyperimmunity, which antibody should you use? There are many antibodies. I hadn't checked every single one, but I guarantee the antibody block interaction between CD20 and CD80 and CD86 definitely work. So that is why you study immunology. Actually, you can, until now, you can see our immune system is so perfect, but they cannot protect virus, cannot protect bacteria, cannot protect fungi. Right. Vaccine is the way we treat immunity, actually. And autoimmunity also, how to say? I think the purpose how we learn immunity is understand the detailed mechanism and how we might have chance to fine tune our immunity to the corresponding disease. It's what I think. Yeah, it's good to know. So any question? I finish. Yeah. I finished 50% of textbook, you understand? I think the first one we have already mentioned. You need T helper cells. Is case, what does that mean? Is case of tuberculosis good or not? Of course, if we can clean the bacteria, it's good. But if the tuberculosis happen, it will not be spread out, so the disease will not also not get bad. Yeah. I think everyone tired. So tomorrow biology will be not so complicated. Yeah, because immunology, how to say? Because for virus, every single virus replicate, in fact, in different way. So we can only talk about something very generally. But for immunology, just this kind of cells. So the only thing we can do is teach you the basic immunology. I wish you can have some. You can learn something. Yeah. What? Skeleton muscle. Skeletal muscle. I think if you do more exercise, your immunity will be higher. Can we finish? I think we can finish now because I need water. So see you tomorrow. 诶，他存档了没啊？他有存吗？他有没有存啊？